Hello friends, today we are going to learn about Agrobacterium tumefacian and how this plant parasite is going to cause crown gall tumor in plants, especially the eudicots, means the monocots, and other than that how we are going to use this plasmid, especially the Ti-plasmid, tumor inducing plasmid in Agrobacterium in recombinant DNA technology for specially transferring genes in plants. Okay, now first of all let's take a look of Agrobacterium tumefacian. Okay, now the biggest challenge in molecular biology is to transfer a gene of interest or the DNA insert any particular gene in plant genome, and not only to transfer but to express that in plant. Okay, so that is the biggest challenge, okay, in front of mole biologists. Now Agrobacterium tumefacian has solved this problem. How? We are going to see that. But Agrobacterium tumefacian is also called Agrobacterium radiobacter, and it is also referred as Rhizobium radiobacter. These are one and the same one. Now, other than that, the optimum temperature for the growth of Agrobacterium is 22 degrees Celsius. But if the temperature will exceed 28 degrees Celsius, this Agrobacterium tumefacian will lost its ability to infect. Okay, especially inside it the tumor inducing plasmid. Okay, definitely the tumor inducing plasmid will show expression, but the proteins which encode the tumor inducing plasmid genes, okay, they will automatically get degraded above 28 degrees Celsius, and that's why above 28 it cannot infect, but 22 is its optimum temperature. This is the best temperature where it can infect and cause crown gall tumor disease to any plant. Now Agrobacterium tumefacian is a soil microbe, gram negative bacilli. Okay, so it's a bacilli, gram negative and soil microbe, and inside it the speciality of it is the presence of Ti-plasmid, tumor inducing plasmid, which is 200 kilobase. This is the tumor inducing plasmid. Okay, clear? And the main thing is, it is not necessary that Ti-plasmid should be present in each and every Agrobacterium. There might be many Agrobacterium in which Ti-plasmid is not present, but if Ti-plasmid is not present, they are unable to infect. Okay, now this Ti-plasmid is going to cause crown gall tumor. Gall, gall means in Greek it means swelling. Okay, and after infection this swelling will go on increasing in a circular manner, and that's why it is called crown gall tumor. Okay, now the wounded plant cells, if we injured a part of the plant, either root, stem, okay, or even leaves, these wounded plant cells, they secrete acetosyringone and some other substances related to it. Okay, this acetosyringone, which is secreted by wounded plant cells, because of that the Agrobacterium tumefacian will get attracted towards that, and inside that, just now what we have seen, that in Agrobacterium tumefacian there is a Ti-plasmid. Ti means tumor inducing plasmid, and in the tumor inducing plasmid there is a Tdna region. Okay, in the Ti-plasmid there is a region called Tdna. It's not tRNA. Okay, clear? Transfer RNA is different. Okay, but this is Tdna, transfer DNA, because this is the left part of or left repeat of 25 base pair, and this is the right, and inside it this region is the Tdna, which is approximately 23 kilobase. Okay, and now this 23 kilobase pair region is removed from the Ti-plasmid and that is transferred in the plant genome. plant genome, it will get inserted in an area which is generally a highly expressible region. इसके कारण क्या होगा? Because of this, as this part contains genes for auxin synthesis, ये Ti-plasmid है, okay, and inside it there is a genes for auxin synthesis, okay. There are genes for auxin synthesis, there are genes for cytokinine synthesis, there are genes for opine synthesis. Auxin, okay, is such a plant hormone which stimulates the cells for continuous cell division. But this auxin synthesis, okay, the number of genes present here which will cause auxin synthesis, the pathway which is followed for auxin synthesis is quite different. Ye pathway, plants are not having this pathway, and that's why plants can't control this auxin synthesis. Similarly, cytokinine synthesis gene is there. Auxin, cytokinine, both are required for cell division. Opine. Opine is an amino acid derivative. Okay, and there are various types of amino acids are there. Opines are there. There are nucleine. Okay, then there are octopine. There are agropine. All these are the types of opines. Okay, and these opine synthesis genes are present in this area. Okay, now this amino acid derivative opine which is the amino acid derivative, can be used as a source of energy, as a source of carbon, as a source of nitrogen. Okay, but the problem with this Tdna region in which there is auxin synthesis genes, cytokinine synthesis genes, and opine synthesis genes are there. This Tdna region cannot get expressed in Agrobacterium. These genes, if Agrobacterium want to express these genes, these three set of genes will be expressed in plant genome, and that's why it is extremely essential to transfer this Tdna region, okay, from Ti-plasmid to the plant genome. And in plant genome, as soon as it will enter in the plant genome, okay, it will get expressed. So auxin synthesis takes place, and as I have explained, this auxin synthesis cannot be controlled by plants. Plant cells इसको control नहीं कर सकते. Cytokinine synthesis will take place, and that's why they will stimulate the cell division, and that's why these plant cells will keep on showing cell division. Clear? Because of that, the tumor is formed, called crown gall tumor. Okay? So this is how it is going to infect. So again, if plant cells will get injured, wounded plant cells, they secrete acetosyringone and various related substances. These attract Agrobacterium towards the area of infection. Agrobacterium is present in the soil, so it can very well travel in that area where there is acetosyringone. And acetosyringone, once it is present, because of that, this Tdna region will get removed, and copy of the Tdna is integrated in plant genome, and here it is expressed. And that's why such plant cells which are having copy of the Tdna will form more in number, forming a tumor. Okay, and all will keep on synthesizing auxin, cytokinine as well as opine, or opine जैसे बनेगा. This opine will be used as a source of energy by Agrobacterium, and now Agrobacterium will start forming its colonies. So what is this? This is actually a type of infection. Okay, but the motto is not to kill the plant. The motto is to grow own colonies. किसके colonies? Agrobacterium tumefacian. Okay, let's take a look of the Ti-plasmid. Okay, we have just seen. Clear? This is the left part and this is the right part. Okay, these are the 225 base pair repeats. Okay, and these regions are going to cut, and this Tdna region, okay, is going to enter the plant genome. But other than that, there are genes for opine catabolism. Means only ओपाइन का सिंथेसिस होना चाहिए. वंस ओपाइन का सिंथेसिस हुआ तो उसको कैटाबोलाइज कैसे करना, उसका यूज कैसे करना एज अ सोर्स ऑफ एनर्जी, कार्बन एंड नाइट्रोजन, दैट जीन्स आर ऑलरेडी देयर. देयर इज अ ओराई, okay, origin of replication, and because of that it's a relaxed mode of replication. Relaxed mode of replication मतलब ये plasmid can form many of its copies inside Agrobacterium. So though we are showing only a single Ti-plasmid, but there must be more than 10 or 20 copies of Ti-plasmid. Okay? Now because the ora is there and relaxed mode of replication, it's not stringent. ये dependent नहीं है. Okay, independent, and that's why it will form its own copies. Then there are, this is a very important region called Vir genes. Vir for virulence. Okay, I can also call it Vir operon. Why? Because it's not again a single gene, there are a set of genes. Okay, and in that, okay, there are many genes like Vir D1, D2, which form endonuclease, and that's why they are mainly responsible for virulence. Okay, because ये Vir D1 and D2 are going to form endonuclease, और यही endonuclease is दो 25 base pair के region को कट करेगा, and after that these two regions will get cut, this Tdna will get transferred. Now keep in mind, there are many Ti-plasmids. Agrobacterium is not going to transfer all. It will only pass few Tdna copies in plant genome. Which plant cells? Those plant cells which are injured. Okay? In there. Now, Vir A is, it is going to recognize acetosyringone and it is present in the plasma membrane. So, this Vir A product, okay, it is present in the plasma membrane and it is going to recognize what? Acetosyringone. Okay, and that's why, clear? Accordingly, it will pass a signal. Then, Vir E, it protects the Tdna. This Tdna region is going to get transferred from Agrobacterium to plant genome, but plants can synthesize their own nucleases to kill, to lyse this Tdna. And that is protected by the Vir E. Similarly, Vir C is there, which will stimulate the transfer in plant genome. Some more Vir genes are there. I have written only the main. Okay, but all these Vir genes are present in these regions. Okay, virulence region. Okay, or I can call virulence operon. This is about what? Ti-plasmid. Okay, so whenever plants get damaged, they will secrete acetosyringone that will attract Agrobacterium tumefacian towards it. The Ti-plasmid present in it, okay, that will, because of acetosyringone, the Vir genes will be expressed. One of the product of Vir genes is endonuclease, which will cut these two regions. Okay, and as these two regions are removed, 25 base pair repeats are removed, this Tdna copy is transferred in plant genome. This is how plants will be stimulated and auxin synthesis will take place, which is not controlled by plants, and that's why it will form tumor. And not only that, but opine synthesis will also take place. They will use opine and they will keep on growing their own colonies. Okay, so this is the mode of infection of Agrobacterium. But today we are going to learn this topic because we want to see its application in gene transfer. Okay, what we have done, we have removed the Tdna region from Ti-plasmid. Okay, the left base pair region and the right base pair. Okay, these two regions are removed, means this Tdna region is removed. Okay, and inside it there must be genes for auxin synthesis, there must be genes for cytokinine synthesis, there must be genes for opine synthesis. Correct? But with the help of restriction endonuclease, we cut inside, remove this portion and introduce a gene of interest and kanamycin resistance gene. Kanamycin is such an antibiotic which can kill prokaryotes as well as eukaryotes. That's why we are using kanamycin here. Okay, and this kanamycin resistance gene is going to act as a marker for us. Okay, so we have gene of interest, we have kanamycin resistance gene, and we fuse them, and this complete fragment of DNA is introduced in this place, replacing auxin, cytokinine and opine synthesis genes. Okay, so you have these two repeats, 25 repeats as it is, and you have gene of interest. There might be more than one gene of interest. I can even transfer an operon there. Okay, and this kanamycin resistance gene. We will require a vector. It's not necessary that you should take only Ti-plasmid. You can take any other plasmid also. Even you can go for Ti-plasmid also. Okay, so I will take a Ti-plasmid, cut in these regions, and exactly this region will be introduced inside it. Or I can take any other expression plasmid, okay, which will act as a vector. Uske baad ligase treatment, and what will happen after that? I will have two types of plasmids. One which are non-recombinant, those plasmids which get closed as it is, but those plasmids which will remain open for more interval of time, and in between them are transferred these fragment. Which fragment? With gene of interest and kanamycin resistance. This is recombinant. Recombinant plasmid will be a bit bigger than non-recombinant because it has genes for gene of interest as well as kanamycin resistance. Correct? And now this mixture of recombinant and non-recombinant is used to transform Agrobacterium. So we have culture of Agrobacterium, and here in the culture of Agrobacterium we have transferred both recombinant plasmid as well as non-recombinant plasmid. Okay, and now some of the Agrobacterium tumefacian will get transformed, but maximum Agrobacterium do not get transformed. So those which are non-transformed will have only their own genome. Correct? These are non-transformed Agrobacterium tumefacian. There might be some Agrobacterium which are transformed but with non-recombinant plasmid. So transformed but with non-recombinant plasmid, and result of this is they will have plasmid but they won't have kanamycin resistance gene, they won't have gene of interest, but very small proportion of the population will have what? This is your genome, Agrobacterium's genome. This is the Ti-plasmid which is originally present in the Agrobacterium. Of course the Ti-plasmid will be present, clear? But this one is having a recombinant plasmid with gene of interest and kanamycin resistance gene. Okay, and now such Agrobacterium tumefacian which are transformed with recombinant plasmid, these we need to transfer in plants. So what we have done, you can see here, this is a leaf and I have prepared explants of the leaves, and I am going to introduce these explants on a medium with growth hormones and kanamycin, clear, which is present there. Okay, so what do we have is a petri plate in which there are Agrobacterium, three types of Agrobacterium. One non-transformed, genome plasmid receiving echia. Second, transformed with non-recombinant, but they will not have kanamycin resistance. These will also not have kanamycin resistance. Only these transformed with recombinant will have kanamycin resistance, and that's why kanamycin which is present in the medium will kill all other, either which are non-transformed will be killed and transformed with non-recombinant will be killed, but the only one which can survive, the only type which can survive are transformed with recombinant. And these bacteria are made to invade the leaf explant, and the leaf explants on a medium with growth hormone. No doubt, growth hormones are required for converting the explant into callus and callus into plant. But kanamycin is also there, and then that's why only those will grow. You can see only this one is growing. Why? Because that must be having what? That must be having Tdna. Okay, clear? Why? Because these are the injured plant cells. They will secrete acetosyringone, and this Agrobacterium will transfer its Tdna region. Okay, at least out of 100 explants, at least one explant will show this transformation. At least one. Okay, and once this recombinant, this region is transferred, that will have kanamycin resistance, not the others. Okay, and that's why this plant is growing. This is the plant which is having gene of interest as well as kanamycin resistance. So, from this plant, many explants can be prepared, and through micropropagation, okay, this plant can be grown, okay, and its after effects of the gene of interest can be studied. Okay, so this is how Agrobacterium is used for transferring gene through recombinant DNA technology. Okay, and this is how novel varieties can be formed. Okay, BT cotton is one of such a variety. Okay, now again we have isolated the Ti-plasmid, the Tdna region from Ti-plasmid, which is having auxin, cytokinine and opine synthesis gene. We replace these with gene of interest and kanamycin resistance. Transfer this in plasmid. Plasmid is transformed in Agrobacterium, and Agrobacterium is made to infect the leaf explant because these are injured plant cells, so definitely they will infect. Okay, and even a single explant will get transformed with recombinant region, which will have gene of interest and kanamycin resistance. Definitely it can form a plant, and from this plant, okay, agronomic trials can be done. Okay, and this is how the Agrobacterium can be used for transferring genes. Okay, so this is all about Agrobacterium tumefacian and its application in gene transfer. Thank you.